So with that, I want to go ahead and introduce our speaker, Dr. Elizabeth Gleim. She received her BA in biology from Hollins University. And prior to her graduate work, Liz spent 2 years working at the CDC, the Centers for Disease Control and Prevention, in the Division of Parasitic Diseases. And then went on after that to get her PhD in wildlife ecology and management with a focus on disease ecology from the Warnell School of Forestry and Natural Resources at the University of Georgia. At Georgia, her research focused on the impacts of long-term prescribed fire on tick population and tick-borne disease risk. Following her doctoral work, she was a visiting faculty member in the biology department at Oxford College of Emory University, and has been back at Hollins, her alma mater, since 2016 as an assistant professor of biology and environmental studies. At Hollins, Liz research continues to be about forest pest and disease ecology, with a particular focus on tick-borne ecology. So we're really glad to have Dr. Gleim here this afternoon to talk with you about ticks and tick-borne diseases. And so with that, I'm going to turn it over to Liz. It's all yours.
Awesome. Well, thanks everybody for taking the time to come and join me this afternoon to learn a little bit about ticks and tick-borne diseases here in Virginia. It's my favorite topic, so I'm excited to share with you some of the happenings here in the state and a little bit more specifically about some of the findings of my research. So just to give you a little overview of sort of what to expect for the presentation today, I'm going to start with just some tick basics, just to sort of get an overview of their life cycle and what the common species are here in Virginia. And then I'll talk a little bit about some of what I'm calling the interesting tick happenings here in Virginia. There's been a number of sort of interesting developments here in the state in the last couple of years relevant to tick-borne diseases. So I'll share those with you all. And that should be a good segue into talking about Lyme disease ecology here in Virginia. It's a really, really interesting state to be in for Lyme disease research, and you'll get to hear more about why that is shortly. And then I wanted to talk a little bit just about how to protect yourself. I'm sure everybody knows at least some of the ways that one can protect yourself, but hopefully I can sort of add to that or give you a few more tools in your handbag to protect yourself, as well as your family members and your pets as well. And then lastly, I'm going to talk about the impacts of prescribed fire on ticks and tick-borne disease risk as a way to potentially reduce disease risk. So whenever I have the opportunity to speak with people about ticks and tick-borne diseases, everyone always has a story, right? Because almost everybody has encountered ticks, and unfortunately, many people have either been personally affected by tick-borne diseases or know someone who has been. So everybody's got some basic knowledge about ticks, right? Especially you all, because you're outside so often, right? But there's also a lot of really common misunderstandings or misperceptions about ticks. And so it's good to kind of get a baseline and see where everybody's at in terms of their own knowledge of ticks and tick-borne diseases. So I thought we might start today with a few quick poll questions to sort of test your knowledge on ticks and tick-borne diseases. So I'm going to ask Chris to go ahead and launch a poll for you all. That I think is just 4 questions about ticks and tick-borne diseases. And if you want to just take a few minutes, or maybe not a few minutes, maybe like a minute or two to fill this out, and then we'll see what the results are, and I'll go over what the answers are. So I'm going to give y'all maybe about, let's say a minute to a minute and a half.
Liz, they can't, I guess they can't see the rash. Let me see if does that help if I move that
Oh,
over?
I
There was a rash.
can, you know what? I'll forward to that one on here, and then hopefully y'all can see that. Hopefully, I'm also probably showing you answers, but oh, I think I went by it. Sorry,
I think
y'all.
you went, there you go. There you go.
Sorry, sort of showed all the answers at the same time, but
Okay.
we'll see.
Hopefully they're able to see, everybody's able to see the questions. I've got a few that are asking where are the questions. Hopefully you should see it. I know a lot of people are voting right now. It should show up somewhere on your screen.
Give everybody else about 30 seconds if you haven't yet been able to finish the questions.
Yeah, they're still coming in, Liz. We've got
Okay.
people are answering. So we've been doing it. It's been out there for about 2 minutes. So you want to give it another till 3 minutes? We'll give them 3 minutes total.
Sure.
So another
Sounds
45
good.
seconds.
Mhm.
I'll give you the answers that we've gotten so far. Okay, well, it seems like there's several people that are saying they can't see the rash for some reason. So, well, that's okay. When we go over the questions, you'll be able to see it, and it's
Okay.
no big deal. We'll just, I'm going to go ahead and end the poll right now. Hold on just a second. Okay, and Liz, let me go ahead and give you. I'll give you some of the answers that we got. So the first question was ticks cannot jump. We got 38% said true, and 62% said false. You want to take these one at a time, or?
Yeah, that works. All right, so so it looks like 38% said true, 62% said false, and the answer there is true, actually. Ticks cannot jump. That's a common misconception that they can jump or fly. Ticks are actually incredibly clumsy. They don't move particularly quickly, and they cannot jump or fly. So oftentimes if people are finding ticks, on their necks or their head, that's usually because they have crawled from your legs typically up to your upper body. Because typically ticks don't tend to hang out in the vegetation any higher than about 1 meter high, and that's pretty high for a tick. Normally they're much lower than that. So that's kind of some good news in some ways, that they're not going to be jumping or flying on you. So that's the first one. Chris, do you want to tell us the results of the next question?
All right. The next question is, what is the most common tick species in Virginia? And 62% said black-legged tick, or the deer tick. 17% said the American dog tick. 20% said the Lone Star tick, and 2% said the Gulf Coast tick.
Awesome. All right, so yeah, it sounds like by far people feel like the most common tick is the black-legged tick, which some people may know more commonly as the deer tick, which is technically not the formal common name, but a lot of people do know it as that. And it turns out the answer is actually C, the Lone Star tick. The Lone Star tick is actually by far the most common tick throughout actually the entire southeastern United States. and it is rapidly migrating up into the northeastern United States as well. And so, yeah, so Lone Star tick's by far the most common. It's incredibly abundant and also tends to seek out humans rather aggressively. And then the second most common tick would be the black-legged tick, followed by American dog tick, and then less common is the Gulf Coast tick, which, as the name implies, tends to only occur in the coastal regions of the southeastern United States.
All
All right,
right.
Chris, next
So
question.
this is the one with the rash that some people couldn't see, but now we can see it. The individual with the below rash, the pictured rash, had what disease? 53% said Lyme disease. 19% said Southern tick-associated rash illness. 14% said Rocky Mountain spotted fever. 14% said this type of rash is not associated with any tick-borne disease.
Awesome. Yeah, so when people see this rash, which is oftentimes called a bullseye rash, and I guess the formal term would be polyurethema migrans, it is classically associated with Lyme disease. Lyme disease is associated with a bullseye rash. But few people know it's also associated with another tick-borne disease called the Southern Tick-Associated Rash Illness, or STAR-I. This disease is actually associated with the Lone Star tick, and we believe that it may actually be often misdiagnosed as Lyme disease because of this really, because of the bullseye rash, right? The good news is that STAR-I is also easily treatable with antibiotics and doesn't have any of the long-term sort of chronic symptoms that sometimes have been associated with Lyme disease. So that's sort of one piece of good news about STAR-I. But yeah, I think it's important to know that this rash is associated with Lyme disease, but can also be associated with STAR-I as well. Rocky Mountain spotted fever is also associated with the rash, but it looks different from this. It's typically more of a diffuse rash that sort of has these sort of red sort of dots sort of throughout or over the body. So also tick-borne disease that's associated with the rash, but looks very different from this bullseye rash here. All right, Chris, and then I think this was our last question. You wanted to go over that one.
It is. Let me do this. For some reason, Liz, some people are saying that they cannot see their slides. So could you stop sharing, then share again, possibly?
Sure.
And see if that helps, and then get back to that slide.
Mm-hmm.
A lot of people are seeing just the title slide for some reason. It might have had something to do with the poll.
If y'all want to let us know if you can see the slides now, that would be fantastic.
Yeah,
hopefully you're seeing a slide now that has that, generally speaking, how long do ticks have to be attached to transmit a causative agent of Lyme disease?
That's right. And so with this question, how long do ticks have to be attached to transmit the causative agent of Lyme disease? 8% said it's immediately transferred. 26% said it's 1 to 2 hours. 17% said 12 hours. And 49% said 24 to 48 hours. And Liz, before I mute myself, maybe after you answer this, maybe you could show them the picture of the rash, and some of them didn't see
Sure.
that. Okay.
Mm-hmm. Yeah, here, let me do that now before I forget. So get that back. There we go. Yeah, so this is the rash that is associated with STAR-I. It's also associated with Lyme disease. We oftentimes call it a bullseye rash because you can see here at the very center would be where the tick had actually been attached. And then it creates this sort of unusual bullseye effect around the bite. So yeah, so that's a classic sign associated with Lyme disease. Not all people who are diagnosed with Lyme disease get a bullseye rash, but most estimates are that 70 to 80% of Lyme disease patients do report a bullseye rash. But this rash is also associated with star eye as well. Okay, so get back to this question. It sounds like the majority of folks were thinking that it was around 24 to 48 hours, and that's correct. So one of the sort of, I guess, good things, if you can say that, about Borrelia burgdorferi, which is the pathogen that causes Lyme disease, is that the tick has to be attached to you for at least 24 to 48 hours to successfully transmit the pathogen. And that's good news because it means that if you remove the tick before that 24-hour time window, you are very likely to not get the disease. So that's a good thing. So I always say probably your best defense against any sort of tick-borne disease is to check yourself, do a full body tick check immediately after you return from outside. And promptly removing any ticks that you find on yourself, because most tick-borne diseases require about 24 to 48 hours for the tick to be attached in order to transmit. There are a few tick-borne diseases that require less time than that, but most require about 24 hours. So doing that tick check and doing it as soon as you can really does a lot to protect you against tick-borne diseases. All right, so the last thing I'll real quickly kind of bring up sort of in terms of basics of tick-borne diseases is I think most people know Lyme disease and many probably know Rocky Mountain spotted fever, but a lot of folks aren't necessarily familiar with the other tick-borne diseases that occur here in the United States and might be surprised to know that there's actually 12 different human tick-borne diseases that are currently established here in the United States. And most of these can occur here in Virginia. And in terms of the most common human biting ticks here in Virginia, there's 4 main species, right? So the first one, as I mentioned, that's the most common is the Lone Star tick. And each tick species typically is able to vector 1 or more pathogens. And in the case of the Lone Star tick, you can see here that they are capable of transmitting a number of different Ehrlichia species, which cause varying forms of ehrlichiosis, and also are associated with the Southern Tick-Associated Rash Illness, or STAR eye. Scientists still aren't entirely sure what the actual pathogen is that's associated with star eye, but we do know that whatever the pathogen is, that it is vectored by the Lone Star tick. The Gulf Coast tick is also here in Virginia, although it really only occurs in the coastal regions of Virginia. And it's associated with a disease called Rickettsia parkeri rickettsioses. Not a great disease name, but nevertheless important to know that it does vector something. A lot of people are familiar with the American dog tick, which is associated with Rocky Mountain spotted fever, and probably the tick species that people are most familiar with is the black-legged tick, which is associated with Lyme disease, as well as a number of other tick-borne diseases, one of which is human granulocytic anaplasmosis, also babesiosis, and so the list goes on with the black-legged tick. And just one other quick sort of piece about ticks in general before we dive into more specific things here in Virginia is the life cycle of ticks. All human biting ticks have 3 main life stages, which are larvae, nymphs, and adults. And typically their life cycle lasts about a year, although in some regions of the US it can be extended for 2 years. And basically what happens is the adult female will lay a clutch of eggs, and typically that clutch will contain hundreds, if not thousands, of eggs. And those eggs will then hatch into larvae, usually sometime in the summer. Those larvae have to have just one single blood meal, and typically it's on something smaller like a bird or a rodent. Once they have fed, they will then drop off into the leaf litter, where they will then molt into nymphs, which are typically active in the springtime and sometimes on into early to midsummer. The nymphs, again, have to have just one single blood meal. Usually it's on something smaller, again, can be rodents, birds, can also be medium-sized mammals like raccoons or possums or things like that. They then drop off into the leaf litter and molt into adults, which again need just that single blood meal, which is usually on something larger like a white-tailed deer, again, dogs, cats, raccoons, etc. And then will breed while on the host. The female will then fall off and lay her eggs in the leaf litter, and then the whole life cycle starts again. Importantly, all life stages theoretically are capable of feeding on humans. But what's interesting is, depending on the species and the life stage of that species, some life stages of the species will be more likely to feed on humans than others. For example, Lone Star ticks and black-legged ticks, typically all life stages will feed on humans or can feed on humans. American dog ticks, for example, typically only adults will feed on humans. Okay, so kind of switching gears, I thought I might talk to you all briefly about some of the sort of interesting tick happenings that have occurred here in the state in the last couple of years. The first sort of interesting piece of news is that we do have a newly emerging tick-borne disease called Borrelia miyamotoi. I'll talk a little bit more about that in just a moment. The other big thing is that we have a new invasive tick species, which some of you all may have heard about, called the Asian longhorn tick. And there's been a lot of news about that because there are some really important implications for both cattle health as well as human health. And Lyme disease dynamics here in the state are constantly changing, and we'll talk more about that as well. So let's first talk about this first piece, which is Borrelia miyamotoi. So Borrelia miyamotoi has actually been around for quite a while, but for many, many, many years, it really was not thought to be able to cause disease in humans. However, that all changed in 2011 when we had our first ever case in humans occur in Russia. And once that case was reported, people all around the world began to look and see if there may be human cases in their own homes, essentially, right? So we knew that Borrelia miyamotoi occurred here in the US, and so scientists and physicians began to look and see if we may have had some undetected cases of Borrelia miyamotoi in the past here in the United States. And in 2012 was the first reported case of Borrelia miyamotoi in a human up in New England. Borrelia miyamotoi is vectored by the black-legged tick, so that's the same tick that also vectors the causative agent of Lyme disease. Since then, we've begun looking for Borrelia miyamotoi in ticks here in Virginia to see if the pathogen is here. And it has been found at a fairly low prevalence, usually less than or around 1% of black-legged ticks have been found to carry this pathogen here in Virginia. I've actually tested for this pathogen myself on ticks here in southwestern Virginia, particularly in the Roanoke area, and found a very low prevalence. Only about 0.3% of the ticks that we tested carried it. And the general thought is that it probably occurs everywhere that the black-legged tick occurs. And the black-legged tick is pretty much distributed throughout all of the entire eastern United States. In terms of the disease, it has flu-like symptoms, so fever, fatigue, those usual sorts of symptoms that you might associate with the flu. And there's no rash associated with this particular disease. So it is treatable by antibiotics, so that's the good news. Initial case reports have indicated that symptoms can be severe enough to require hospitalization, but it's not entirely clear whether the disease is really that serious or if it's just that the patients that actually are getting diagnosed are the ones that are going to the hospital, right? Because that's when you start to have more advanced diagnostics happening for people, where it's entirely possible that many people may be experiencing milder versions of this Borrelia myomycotoii infection that are just chalking it up to be a virus or a flu, right? And never get, never see a doctor, or maybe go to the doctor and the doctor just says, oh, it's a virus, go home and don't worry about it, right? So still very much a lot to be learned about this particular pathogen, but important to know that it is here in the state. I also mentioned this new non-native invasive tick species called the Asian longhorn tick. And you all may have seen news about this in the last couple of years here in Virginia. And one of the things that's particularly concerning about this tick, and the reason that it's considered to be invasive, is because, as this headline says here, it's capable of essentially cloning itself. It undergoes what we call parthenogenesis, which means that the female does not have to breed with a male in order to lay eggs, that she can just simply lay eggs. And so basically all of her offspring are clones of themselves. And interestingly enough, the entire population here in the US are all females because of that. So a little bit of information about the Asian longhorn tick. It's indigenous to Eastern Asia and is distributed throughout much of the world now, either because of, in terms of Eastern Asia, it's native to there, and it has since been distributed throughout many other places around the world. And in those other places, it is a human and animal disease agent. It's capable of vectoring the pathogens that cause severe fever with thrombocytopenia syndrome virus, long one, long name there, Japanese spotted fever, and is also a potential vector, or is known to be a vector in other places around the world for pathogens that are in the same genus as pathogens that we have here in the United States. It was first detected here in the US in August of 2017 in New Jersey on a sheep farm. At the time, people weren't especially concerned. They said, hey, you know, this is a tick that's indigenous to Eastern Asia. It typically does better in more tropical climates. And surely these ticks are going to be killed off in the winter in New Jersey, right? That was the thought anyway. Unfortunately, winter came and went, and in the spring of 2018, tick scientists went out and started looking to see if the tick was still present in the environment there around the farm, and they realized that it was still there. And that's when we realized that we had a problem. So at that point, people began looking for the tick throughout New Jersey, and they began finding that it was more widely distributed than they had expected. And then we started widening our search lens and looking elsewhere throughout the Eastern US. And we now know that it is distributed throughout now, I guess, 12 different states in the Eastern United States. And anywhere you see blue is a county where the tick has been detected. And this is an older map, but it's the most recent one I can find from 2018, the end of 2018. And what we realized, you might be thinking, how did we not find this tick before, right? If it was so widely distributed. Well, it turns out that we have so the scientific name of this tick is Hemophysalis longicornis. And it turns out that we have another Hemophysalis species here in the United States called Hemophysalis leporus palustris. And those ticks look really, really similar to one another. And what we realized as we began, actually people started going back and looking at their stored collections of ticks, right, at universities and museums. and reevaluating some of these ticks that we had been identifying as this native Hemophysalis species, and we call that the rabbit tick. And we realized, oh, those had been misidentified, and in fact, some of those were this Asian longhorn tick. We know now, right, as you can see from this map, that it's here in Virginia. It is widely distributed throughout the western portion of Virginia. Everything that we can see so far from sampling indicates that it's not yet present along the coast in sort of central Virginia, but is almost everywhere along western Virginia, with a few exceptions right around here in southwestern Virginia, ironically around the Roanoke Valley area where I live. It hasn't quite made it to us yet for some reason. But one of the things that is particularly concerning about this tick being here in the United States is, number one, we are not sure whether it's capable of vectoring any of those tick-borne pathogens that we have that are native here to the United States. For example, Anaplasma, Borrelia, Babesia, Ehrlichia, Rickettsia, these are all genre of tick-borne pathogens that occur here in the US. There are other species in the same genre elsewhere in the world that we know it can vector those species. So the question is, can it vector the specific species that we have here in the US? Research is ongoing with that. And what we know so far is that it has been found to be a competent vector for the causative agent of Rocky Mountain spotted fever. Initial research looking at whether it could possibly vector the bacteria that causes Lyme disease indicates that it probably doesn't, but we still need to do a little bit of work with that. And no other tick-borne pathogens have been studied yet to see if the Asian longhorn tick could potentially vector those as well. One other thing we know that it also is capable of vectoring is a type of tick-borne pathogen that actually affects cattle, and that has been confirmed here in the US. And typically that tick-borne disease in cattle is usually not going to cause death of the cattle, but particularly for dairy cattle, it's an issue because it'll actually cause a reduction in milk production, which is obviously a really significant issue for dairy farmers. So there's a lot of concern around that as well. So it was, and I guess I should go back and just quickly mention, the Asian longhorn tick was first detected here in Virginia back in the summer of 2018, up around Charlottesville. And as we see, right, it's now sort of distributed throughout much of Western Virginia, or I shouldn't say distributed, more like has been detected. It's probably been there for a while. And I'll just reiterate, if anybody has any questions at any point, feel free to type the question in the Q&A.
Well, Liz, let me break in for a second. Sorry about that.
Sure. Mm-hmm.
What I'm going to do, we've had some issues with some sound and the slides advancing, and so we're trying to work with that. And
oh no.
Devin has told us that sometimes leaving the meeting then coming back will fix that. And I think some people have had luck with that, but some haven't. So I think there have been some problems with the last few days with iPads and iPhones, and things look differently. So we'll ask everybody to maybe, if they're having issues with that, if you try logging off and logging back on, it might work for you. And Liz, I was also going to say, because of all these problems, I do have a lot of questions, but I think because of that, I'm going to save them till the end. Is that all right? Because we got
that's fine.
Built up, and there's a backlog of them now. So when you finish, we'll start the questions. How's that?
Sure. Yeah, that sounds great.
Thanks.
Awesome. Okay. Okay, so kind of transitioning now to talk about Lyme disease a little bit. So a little bit of basics of Lyme disease. It is the most common vector-borne disease in North America, including, of course, most common vector-borne disease here in the US. 30,000 cases are reported to the CDC annually, but it's actually estimated that the actual number of cases that are probably really occurring in the United States is likely closer to 300,000. And this disease is caused by a pathogen, particularly, in particular, a bacteria called Borrelia burgdorferi. Cases in the U.S. have been increasing pretty steadily over the last several decades. And I'll talk a little bit about sort of some of the postulated ideas for why that is a little bit later. And the vector is actually several different Ixodes species here in the US. But the most common one, right, that's really associated with human transmission is Ixodes scapularis, the black-legged tick that we've mentioned before. There are a few other tick species, for example, Ixodes affinis, that don't really feed on humans, but we think maybe playing a role in maintaining the pathogen in the wild. But Ixodes scapularis is really the only Ixodes species here in the US that feeds on humans. So they're the ones that are transmitting Borrelia burgdorferi to people. So when we look at where Lyme disease is happening in the US, cases are really concentrated in the northeastern US and in the upper Midwest, which is interesting because if you look at the distribution of the black-legged tick, it's really distributed throughout the entire eastern US. So one of the really big questions among disease ecologists and public health officials is why is it not occurring in the southeast, right? The tick is there, but for some reason the disease is not. And there's been a lot of different hypotheses for why exactly that is, and it's very much an active area of research and has been now for, honestly, probably 1 or 2 decades. And there's 2 main leading hypotheses for why these differences occur. The first is that there may be, well, the first is that there's distinct northern and southern populations of the black-legged tick, and that those populations may be using different wildlife hosts for those blood meals that they need to complete their life cycle. And it turns out that the wildlife hosts that ticks up in the northeastern portion of the US tend to be on white-footed mice and other wildlife hosts that are really, really good reservoirs for the pathogen that causes Lyme disease. Alternatively, the wildlife hosts that ticks tend to use in the southeastern US include things like lizards and other wildlife species that are not good reservoirs for the pathogen that causes Lyme disease. And so the ticks, therefore, would not be becoming infected with Borrelia burgdorferi. So that's one hypothesis for why Lyme disease is common in the north and less common in the south. The other hypothesis that's been getting a lot of traction a little more recently is that I mentioned before that there are genetically distinct northern and southern populations of black-legged ticks. And there's evidence that the northern population of black-legged ticks has much more aggressive questing behavior than the southern ticks do. What is questing? Well, when ticks have to find a host, right, they do that by oftentimes detecting the CO2 that we and other animals are breathing out, among other things. And when they do that, they will crawl up to the top of any vegetation in the area. So things like grass or low-lying herbaceous plants on the forest floor, and they will wait and hope that that wildlife host physically brushes up against them as they're walking by. And with black-legged ticks in the north, it's been found that that questing behavior is more aggressive, meaning that they're more likely to crawl higher on the vegetation in order to find a host. Whereas the black-legged ticks in the south are not very aggressive, particularly the nymphs are not very aggressive. And they actually stay down in the leaf litter and seem to find hosts that are sort of moving through the leaf litter. What that means is that if they're staying down in the leaf litter, they're not getting on people. And if they're crawling up on the tips of vegetation like they do up in the Northeast, then they are more likely to get on a person. So that's one of the other big hypotheses, or why we have the difference in Lyme disease from the Northeast versus the Southeast. Now, when we start looking at spatial trends of Lyme disease and sort of how it's spread over the last couple of decades, we can see that back in 2001, it was primarily concentrated in the northeastern US, and since then has had a pretty significant spatial sort of expansion throughout the northeastern US and the upper Midwest. But if we really focus in on Virginia, you can see some really interesting, from a disease ecologist perspective, things happening. In 2001, you can see that there were very few cases of Lyme disease in the state at that time. But if we fast forward, let me take that away actually, if we fast forward to 2017, we can see that this sort of southern front of Lyme disease seems to be migrating further southward. And in particular, Lyme cases in Virginia seem to be concentrating along the western portion of the state, with this hotspot forming in southwestern Virginia, particularly in the Roanoke-Blacksburg area, which just happens to be where I live. So if we zoom in a little bit more on what's happening here in Virginia, and I sort of call Virginia this sort of front line of Lyme disease because that southern front is sort of moving down across the state. If we look at Lyme cases in Virginia, which are the white bars here in this graph, you can see that in 2007, cases jumped pretty dramatically, and that since then, cases have continued to increase. And when we look at a map of where those cases seem to be increasing the most, right, this is annual change. So basically mapping, where cases are increasing or decreasing, we can see again that cases are really increasing primarily along western Virginia, particularly in southwestern Virginia here, which is the Roanoke-Blacksburg kind of Floyd area. So why is that, right? That's the big question. What is happening here? Well, there was a study that was done in 2014 by Jory Brinkerhoff at University of Richmond and some of his colleagues. And what they did was they collected black-legged ticks on sort of across an east-west gradient in Virginia. And this is where he collected those ticks, right? So each one of these black dots here is a location where he sampled those ticks. And what he found was that this LE site, his most western site, which you can see here is right in the mountains, was where he found the highest density of black-legged ticks. And those ticks also had the highest prevalence of Borrelia burgdorferi, the causative agent of Lyme disease. And so based off of that, he postulated that we may have higher densities of black-legged ticks here, and that they may be carrying a higher prevalence of the pathogen. He also postulated that it may be that for whatever reason, the black-legged ticks were preferring higher elevations, because again, those Lyme cases seem to basically be following the mountains, which run along the western portion of Virginia here. But right, this was all based off of just a single site in western Virginia. So it really sort of highlighted the need for us to do some more extensive studies here in western Virginia, and ideally, particularly in southwestern Virginia down here, where that hot spot has developed, to really understand what's going on. So, right, why are we seeing these high cases in western Virginia, and especially in the Roanoke Valley and that hot spot that I showed y'all before? As I mentioned before, It could be because of simply just, for whatever reason, we have more black-legged ticks, Ixodes scapularis, in the western portion of the state. It's also possible, as I mentioned previously, that it could be because our ticks have a higher prevalence of Borrelia burgdorferi, the causative agent of Lyme disease, right? But there are other things that could be going on as well. Number one, it's possible that we are simply seeing over-reporting of Lyme disease for whatever reason in this portion of the state. That doesn't seem particularly likely in this particular scenario, but it's always something that you have to consider when you see an area with high case numbers of any disease. You have to ask, is it just that maybe our physicians are more likely to report the disease, even though theoretically all physicians are required to report Lyme, or that they're more likely to diagnose it, etc. Again, that doesn't seem like that's probably the case here, but it's always something we have to consider. It's also possible that we have populations of black-legged ticks that quest more aggressively. In other words, that are showing that northern aggressive questing behavior as opposed to the less aggressive southern questing behavior. And we'll talk more about that in a moment. It's also possible that our black-legged ticks have higher contact rates with humans, right? So regardless of how aggressively they're questing or how many ticks there are, it could be that humans are spending more time in tick habitats and therefore coming into contact with ticks more frequently, right? So that could also be happening here in Western Virginia. And then the other possibility, which in all honesty is probably the most likely possibility, it's really some combination of all of these things. So as I mentioned before, Brinkerhoff et al. Sort of postulated that there's some evidence that there could be greater abundance of black-legged ticks and greater prevalence. But these other hypotheses really had not been explored, and even those that Brinkerhoff had postulated needed further research as well. So with that, I moved up here to Roanoke in 2016, and I was immediately interested in this question and was surprised to find that there really weren't any tick ecologists here in this region doing the work. So when I first started doing research here and establishing my program, we had to start with very basic things, just right, what are the tick species here in southwestern Virginia? How abundant are they? When are they active, right? That's phenology, which is just when are all those different life stages of every different species active within the Roanoke Valley, right? So just to take one quick step back, I mentioned, there's 3 life stages for all human biting ticks, larvae, nymphs, and adults. And every species, and every life stage of every species tends to be active at certain times of the year. And when they're active is different depending on the life stage and the species. So we call that phenology, when all those different life stages and species are active. And it's really important to have an understanding of phenology in different regions of a state, and certainly throughout the country, because that phenology varies from region to region based off of, differences in climate and ecosystems and everything else. And the second thing that we wanted to do was we wanted to determine the prevalence of Borrelia burgdorferi in Ixodes scapularis. So what is the prevalence of this agent that causes Lyme disease in black-legged ticks? Was that high prevalence that Brinkerhoff saw similar to what we are seeing in other places around Western Virginia, or was that just simply a fluke, right? So to do this work, we established 12 different sites in the greater Roanoke Valley area, which you can see sort of fell within this triangle here. And we went on a monthly basis and we collected ticks via flagging, which is what my student Sierra is doing here. You can see we literally use what looks like a flag. And I mentioned that ticks to get on a host will quest up to the tops of the leaf litter or low-lying vegetation and just wait for something to brush up against them. And that's what we do with the flag. The flag just simulates a host brushing up against them and picking them up, and then we collect them. We ultimately, over the course, we did this for a full year, the monthly collections at these 12 sites. We ultimately collected almost 20,000 ticks, so we got a lot of ticks. We brought those ticks back to the lab and we identified them. And then we chose to test the black-legged tick nymphs for Borrelia burgdorferi, and that's what my student Limu is doing here, was doing that pathogen testing for the causative agent of Lyme disease. You might be asking, why did you test nymphs? It turns out that, as you'll recall, like for the life cycle of ticks, there's larvae, nymphs, and adults. Larvae do not carry the causative agent of Lyme disease. Only nymphs and adults do. But it turns out that nymphs are much more highly associated with human cases of Lyme disease than adults are, because they're so small that a lot of times people don't see them and feel them when they're biting. And so they're more likely to be able to stay attached to a person for that 24 to 48 hours because they simply are undetected. Whereas the adults are larger. Oftentimes you feel them crawling on you, or you certainly see them because they're just bigger. And so a lot of times they're not able to stay attached long enough to transmit the pathogen. So nymphs are very relevant because they're most associated with human Lyme disease. So we chose to focus on that particular life stage for testing initially. Okay, so what did we find? Again, we weren't only interested in black-legged ticks, we were interested in all ticks when we were looking at basic phenology and things like that. And generally speaking, we found mostly what we expected, right? So you can see here that our Lone Star tick adults in the solid orange line, and also our American dog ticks in the solid green line, are active primarily from May, June, and July. The black-legged tick adults in the solid blue line are actually out in November, December, and even in January and February, which is what we expect. They tend to be active in the winter, the adults do. But then you see the big peaks, right? So you see our Lone Star nymphs are very active from April all the way through to August. And again, by far the most abundant tick out of all of our ticks. And I mentioned to you all, right, Lone Star ticks are the most abundant tick in the eastern US, so that wasn't surprising to us. But this is what was really interesting. We see our dotted blue line here, and those are our black-legged tick nymphs. I mentioned to you all before, right, that the southern black-legged ticks tend to not quest very aggressively. They tend to stay down in the leaf litter. And if they're staying down the leaf litter, we don't tend to collect them by flagging, which was the method we were using. So the fact that we were catching all these black-legged tick nymphs indicated that they were questing aggressively, like those northern ticks are. And that's unique because it turns out there are and they work in central and eastern Virginia. And what they had shared with me is that they don't tend to collect black-legged nymphs, meaning that they tend to have those southern populations of black-legged ticks that tend to stay down in the leaf litter. So the fact that I was finding a very significant peak in activity in May through July of these nymphs was indicating that we seemed to have black-legged tick nymphs that were showing more northern characteristics. And that implied that what we may be having is ticks from the north, where Lyme disease is common, are migrating down along the Appalachian Mountains and establishing a new population here in Virginia. To sort of further, I guess, explore this idea that we were having very different sort of northern questing behavior versus southern questing behavior, I actually looked at some of my old data from graduate school. So you all might remember in my introduction, Chris mentioned that I did my graduate work at UGA, and specifically worked down in southwestern Georgia for that work, where I collected lots of ticks using this very same method. And what I found was really interesting. I found that when you looked at adults and how many I captured, in southwestern Virginia, I got about 2.3 ticks per hour, adults that is. In southwestern Virginia, I got about 3.4 ticks per hour, right? And when we take in our sort of standard error for those measurements, it basically indicates that these were not significantly different numbers, indicating that the abundance of black-legged ticks in Georgia versus Virginia is not really different. That's interesting, right? Because Lyme disease doesn't really happen in Georgia. But then when we look at what I got for nymphs, in our current study in southwestern Virginia, we were getting about 12 ticks per hour during their peak activity. And I got 0 the entire time I did my work down in southwestern Virginia, or sorry, southwestern Georgia. So that seems to indicate, right, that there is a very different behavior that those ticks, particularly the nymphs, are showing that may be contributing to this high peak of Lyme cases in Western Virginia. And so our other big question, right, was what's the prevalence of Borrelia burgdorferi, the causative agent of Lyme disease in our ticks here in Virginia? In total, we tested 291 nymphs. That was all of the nymphs that we collected. And of those, 3.8%, so 11 of those ticks, were positive for Borrelia burgdorferi, right? So only 3.8% of the nymphs that we collected were positive for the pathogen that causes Lyme disease. You'll note we also got a single tick that was positive for that Borrelia miyamotoi that I mentioned earlier. So this was a little surprising that we found such a low prevalence of Borrelia burgdorferi in our ticks, because in that Brinkerhoff paper that I told you all about before, he had a very high prevalence in that one Western site. He had about 20% prevalence of the pathogen there. The only other study that's been done in Western Virginia on Borrelia burgdorferi prevalence in black-legged ticks was actually done in southwestern Virginia, in the same region we're in, but they only tested adults. And adult ticks tend to have much higher prevalences of pathogens than nymphs do, because they've had one more blood meal, so they've had one more opportunity to get ticks, or sorry, to get the pathogen. So their findings aren't really, we can't really compare ours to theirs, because it's kind of like comparing apples and oranges. But still it's informative, right? So this was interesting because we have this lower prevalence of Borrelia burgdorferi as compared to sort of other studies that sort of aren't directly comparable, but it's the best we've got to work with, right? Okay, so what does this all mean? Number one, it seems like black-legged ticks, Ixodes scapularis, that their nipple activity is similar to ticks from the northeastern US, right? And that does not correlate with the behavior that we have in other regions of the state, which seems to further validate perhaps what might be one of, at least, the driving factors behind the high cases incidence that we have here in Western Virginia. Borrelia burgdorferi prevalence is fairly low, at least according to our findings. And if that is indeed the case throughout Western Virginia, then it would not therefore seem to indicate that high prevalence is a primary contributor to the high Lyme incidence that we have here in this portion of the state. But more work needs to be done in other portions of Western Virginia to really further explore that. The other piece that we've begun to consider is the fact that these Lyme cases are concentrating around the western portion of Virginia in the mountains. And the mountains, we tend to see a lot of outdoor recreational activity, right? And Roanoke in particular is sort of a unique place because we're a small city, and in general, we don't expect to see high case incidences of tick-borne diseases in cities because people aren't really spending much time outdoors, right? But that's not the case here. Roanoke is very much marketed on having this wonderful outdoor recreational lifestyle. We have tons of amazing trails. We're on the Appalachian Trail. We have one of the US's best parks for mountain biking. We have Smith Mountain Lake, right? We have all these amazing places to go and spend time outside, and people very much go and spend time outside. And so what that has made us wonder is if this high Lyme case incidence may be driven by, number one, the more aggressive questing behavior of our black-legged tick nymphs, which may be because ticks are migrating from the north into Western Virginia, in conjunction with a very large human population, particularly here in the Roanoke-Blacksburg area, combined with the fact that this large population of people is spending a lot of time outdoors in tick habitat, right? All of these things we suspect may be driving this high Lyme incidence that we're seeing here in Western Virginia, and more specifically in southwestern Virginia here in the Roanoke-Blacksburg area. So we're still doing some research on this to further explore some of those other hypotheses to see if they too could be contributing to the high Lyme incidence that we have here in southwestern Virginia. But at least the initial findings seem to be indicating that we're at least figuring out part of the puzzle anyway, so that's encouraging. So speaking of Lyme disease, I thought I might really quickly talk about how you can reduce your risk of tick-borne diseases, because being master gardeners, I'm sure that you all are spending a lot of time outside in wonderful tick habitat. So really quickly, just some tips for how to prevent tick-borne disease. Right, your first line of defense is to keep ticks off of you in the first place, right? So to do that, you can use repellents. Particularly, I recommend repellents that include DEET in them. And again, you can spray those on your clothes if you don't want to necessarily spray your skin with them, particularly if you're wearing pants, which I recommend. You can also treat your clothes themselves with things like permethrin. And permethrin is really convenient in that you can treat your clothes once, and that treatment will typically last for about 6 weeks or up to several washes. And I also really strongly encourage folks to consider wearing long pants, even in the summer, when you're out in ideal tick habitat, so out in the woods, out in long grass, things like that. Wear closed-toed shoes. If you can bear it, wear long sleeves. But as I mentioned before, ticks don't tend to quest very high, so they typically get on your pants first or your legs, and then they crawl up. So even if you wear short sleeve shirt but with pants, that's a pretty good defense. I also really strongly encourage folks to wear light colored clothes, especially light colored pants, so that you can more easily see the ticks if they are on you, so that you can sort of remove them before they even find your skin. So, you know, wearing khaki colored pants or white pants or things like that are better than wearing black pants. And the other piece is, you know, to check your clothes occasionally while you're out in the tick habitat, in the woods or wherever it may be. I know for me, I try to occasionally just pause what I'm doing and look down and just check, do I see any ticks crawling on my pants or on my shirt, right? And remove them if I do. If you want to look really cool, you can also tuck your pants into your socks to add an extra layer or barrier, if you will, so that ticks can't, you know, crawl under your pants and find your legs, right? So that can also be really helpful. I also, as we're looking at socks here, I will remind folks, because they often don't think about this, if you are treating your clothes with permethrin, don't forget your socks. Treat your socks as well. When you get home, promptly change your clothes and ideally wash them and shower. This will get any ticks that haven't yet attached to you off. And ticks often take a while to actually attach. So you would be surprised how many ticks you may remove just by simply changing your clothes and washing, taking a shower and washing them away, right? And then the last thing that you can do, and I always tell people, if there was literally only one thing I was allowed to do to protect myself from tick-borne diseases, it would be this. That is a full body tick check as soon as you get back from being outside. So, right, you want to remove any ticks that you find as soon as you find them. I know sometimes people are sort of nervous and I don't know if I can remove it myself. Maybe I'll wait until, you know, my husband or whomever can help me remove it, or a doctor can remove it or whatever, right? But it's really better to go ahead and try to remove it as soon as you find it rather than wait, because as we know, the longer the tick is attached to you, the more likely it is going to be able to successfully transmit a pathogen if it is carrying one. And if you have children or grandchildren, don't forget to check them as well, right? And I'll real quickly also mention sort of what you can do to help protect your pets, because, right, pets are also susceptible to some of these tick-borne diseases, right? Dogs can get Lyme disease. They can also get ehrlichiosis. Horses can as well. So what can you do? Number one, use regular tick preventative, right? So speak with your vet, ask them what they recommend in terms of a tick preventative, and be sure to use it throughout the year, right? Because as I mentioned before, different species and different life stages are active at different times of year. There is no time of year that ticks are not out. You might also, if you have a dog, consider the Lyme vaccine. It doesn't completely protect them from Lyme disease, but it hopefully provides some protection and may make the disease less severe if they were to contract it. You also, of course, want to do just as you would for yourself, for your pet, check them and remove ticks, particularly after they've been out in the woods or in tall grass. And if your pet becomes lethargic, if they have a loss of appetite, if they have inconsistent lameness or swollen joints, all of these can be signs of a tick-borne disease, and you should go and see your vet immediately if this should happen. Okay, so what should you do if you find a tick on you, right? How do you remove them correctly, and what do you do afterwards? So this is just sort of a diagram of some of the do's and don'ts of removing a tick. The first thing is you want to grab them ideally with some fine-tipped tweezers as close to your skin as possible, right? So you want to avoid mushing or mashing the body of the tick. You really want to be grabbing it down here, right at the skin, okay? And then you want to gently pull straight out. Right, so you don't want to twist it and you don't want to jerk it out. You just want to slowly and steadily pull it straight out. Once you've removed the tick, you want to clean the area with soap and water, or hydrogen peroxide rubbing, something to disinfect, right? And then apply some antibiotic ointment. So just treat it like you would any other cut or abrasion, right? You also want to keep the tick, so don't throw it away. The easiest thing you can do is put it in a Ziploc bag and freeze it, as odd as that sounds. If you have ethanol, rubbing alcohol, something like that, you can also preserve the tick in that. And keep the tick for up to a month. And ideally, you want to label it so that you can keep track of what tick came from what day and what person and all that kind of stuff. You want to keep it for a month so that if you were to develop any sort of tick-borne disease, you can have that tick tested. There are places that you can send your ticks where they will test it for you, you know, at a price, but typically I think I've seen prices of like $50 to $80 or something like that, and they'll test it for common tick-borne pathogens. So that can be really helpful if you're trying to diagnose a tick-borne disease, or if you found one and you're concerned that, you know, you're wondering whether you should be concerned about a tick-borne disease, right? So that can be good to keep the tick. The other thing that I do is I check the tick and inspect for the level of engorgement, because how engorged the tick is may help provide some clues for how long the tick has been attached to you. So if you find the tick is engorged at all, it probably indicates that it's been attached to you for at least 24 hours, if not longer. Right, and then you know that you might be a little more concerned because a tick has been attached long enough to transmit a pathogen to you. If the tick is completely flat, I'm never brazen enough to say, you're good, you're fine, right? But you can at least feel a little better that it might appear that the tick has not been attached to you for very long. So chance of transmission of the pathogen is lower. Few other last things I'll mention about tick removal. There's a lot of wives' tales out there about how to safely remove ticks or how to remove ticks. Things like burning the tick, smothering the tick with Vaseline or nail polish remover, any of those sort of things that are out there, those actually may increase the chance of the tick transmitting any pathogen that it's carrying to you, because by you doing that, it's basically increasing the likelihood that it's going to regurgitate into you. And that's how it's transmitting the pathogen, essentially. So no Vaseline, no burning the tick, nothing like that, right? Just simply grabbing the tick as close to your skin as possible and pulling straight out. You all are probably also curious to hear how you might be able to control ticks out in the environment around your home. So if we're talking about areas like landscaped, mowed, maintained areas around your home, right? There's a number of things that you can do to make it a less hospitable place for ticks. Number one, mowing does a lot to basically make the habitat unhospitable to ticks. So if you can mow your grass consistently and keep leaf litter down or completely out of your yard, that's removing a lot of the draw that your yard might have for ticks. You can also do things like create barriers around your property, like creating a 3-foot wood chip barrier or gravel barrier around the perimeter of your property, or potentially if a certain part of your property backs up to woods or some kind of more ideal tick habitat, you might consider putting that wood chip barrier or gravel barrier down there, because the ticks are very unlikely to travel across that barrier on their own. Now, that doesn't mean that a wildlife host isn't going to just cross over that barrier and bring ticks with them, but it may at least reduce the chance of ticks just migrating themselves into your yard. Other things to think about is if you have areas that people are going to frequent, whether that's a play set or a fire pit or a patio, placing those things well away from wooded areas or tall grass, again, will reduce risk of ticks finding you in those areas, right? The last thing I'll mention is some people do use pesticides or caricides, as we call them in the tick world. Obviously, there's concern about pesticides or caricides because typically they are not only affecting ticks, they're also affecting good insects, particularly pollinators. So I am always hesitant to recommend the use of those, but you will certainly see those services being offered by pest control companies. So they're out there. But in my mind, there are many other things, as you can see, that you can do to reduce risk of tick-borne diseases and ticks in your area that wouldn't necessarily be as detrimental to other insects. And what about larger pieces of land, right? Because I'm guessing maybe some of you may own some acreage. And there have been so many studies done examining different tick control measures that can be applied in some instances in residential settings, and some really are more appropriate for like larger wooded areas, right? Prescribed burning has been explored. Obviously not going to happen in your yard, but it can certainly happen in larger wooded areas, or even in areas that may border residential areas. Other things that have been explored are herbicides, so killing low-lying vegetation, sort of similar to mowing in your yard, right? Makes it a less hospitable place for ticks. Acaricides, i.e. pesticides we talked about. Mechanically clearing undergrowth. So, you know, basically roller chopping or whatever it may be, undergrowth in a wooded area. There are a lot of studies now that have actually looked at directly applying acaricides, i.e. pesticides, to white-tailed deer or rodents, which are really important hosts for ticks. And there's been varying levels of success with those. And there's also been some studies that have explored just simply thinning deer populations by hunting or things of that nature. So all of these things, let me say first of all, none of these things have appeared to be like the answer, right? All these have been studied pretty extensively. All of them have tended to have varying results. And many of these, right, some may be more appropriate for residential settings, but not really logistically feasible in larger wooded areas, and vice versa. Some things may be more realistically used in a large wooded area that wouldn't necessarily work in a residential area. So there's a lot to consider when you're looking at control methods. But to me, one of the more promising methods for controlling ticks on a landscape level is prescribed fire. So when we look at studies that have examined prescribed fire, we know a couple of things. We know that tick populations are reduced immediately after fire, but a lot of studies have found that those tick populations will simply recover over time. Usually within a year, they'll return to pre-burn levels. But there's some dispute over the impacts of long-term prescribed fire on tick populations. Some studies have found increases in ticks, some have found decreases, and some have just found that the ticks rebound right back to where they were at before the fire. When I started looking at this body of literature and trying to understand why we had such differing outcomes in these different studies, I found there were a couple sort of things that emerged. Number one, a lot of these studies previously done failed to account for other variables that were known to affect tick populations, like wildlife host abundance, forest structure, etc. And or they didn't simulate real-world management practices, meaning that in the studies, they may have only done a single burn, or they may have burned a really small area of land, maybe just 5 acres. And that's not typically what we see when prescribed fire is used in a real management scenario. Typically, prescribed fire, when it's used by land managers, it's done on a regular basis, typically every 2 to 5 years. And it's done on a much larger scale. Oftentimes, dozens, if not hundreds, and sometimes thousands of acres are burned at a time. And you can imagine that those differences have really major implications for how easy it would be for a tick population to reestablish itself after a burn. So what I did in graduate school was I wanted to look at how does long-term prescribed fire affect tick populations, while accounting for those other variables like hosts, like climate, like forest structure that affect tick populations. So I went and I worked in areas that had been subjected to prescribed fire for at least 10 years. And then I also had some control sites where burning had not occurred for at least 10 years. And what we found was pretty dramatic. You can see here, I collected ticks on a monthly basis for 2 years. And what I have here is average number of ticks infected with pathogenic bacteria that were encountered per hour, okay? So this is how often would you encounter a tick that would actually, if it was able to bite you and, you know, stay attached for at least 24 to 48 hours, actually transmit a tick-borne disease, right? And so you can see at my burned sites, it almost flatlined, right? For many months, there were no ticks at all that were infected with pathogenic bacteria. And in the months where there were some ticks that were present with disease-causing bacteria, there were very low numbers. We're talking like 0.04 ticks an hour, right? Compared to our control sites where burning didn't happen, where you can see normal seasonality of ticks infected with disease-causing bacteria. And we're talking at peak times encountering 1 tick per hour that's infected with a bacteria that can cause disease, right? So ultimately, we're talking about you can only spend an hour in a forest and you may very well encounter a tick that's carrying pathogenic bacteria, versus in these burned areas, you would probably have to spend at least 10 hours or more at these sites before you ever encountered a tick with disease-causing bacteria. So this was really exciting because it really seemed to indicate that long-term prescribed fire does reduce tick populations significantly, regardless of how frequent the burn is, how abundant hosts are, etc. Right? We ultimately documented a 98% reduction in encounter rates with ticks infected with disease-causing bacteria. Right? When we looked at why, we found that the big thing was that the fire is, from a land manager's perspective, is used to mold the landscape, mold the vegetation that's there on the landscape. And that fire basically creates a vegetation structure in the forest that is not hospitable to ticks. It generally creates a hotter, drier environment, and the ticks just can't simply survive in that. And that was a really important finding because what it really emphasized was the need for long-term consistent burning, right? It indicated that singular burns for control of ticks would not be effective, and in many cases may actually be counterproductive. So that was really interesting and really exciting findings that, again, this work was done down in Georgia. And so one of the big questions I now have is, how does this translate in Lyme endemic areas like Virginia? And I hope that we may be able to explore that more in the coming years here. The last thing I'll mention with you all, and I know I'm almost out of time, which I hate because I haven't even been able to answer questions yet. But I want to share this with you all because I think it may be of interest. If you have forested land and you might be thinking, I don't know how to do prescribed burn, I can't do a burn, there are resources for you. Obviously, you cannot just go out and decide to burn your woods. You know, there are laws, there's regulations associated with burning, and it does require training to do a burn safely. But there are resources out there for you. So the Virginia Department of Forestry does offer a 3-day program to become a certified prescribed burn manager. And it's open to anybody, so anyone could do that. There are also a number of private contractors out there who are trained and experienced in doing prescribed burns, whom you can hire to come and burn your land. And the Department of Game and Inland Fisheries, DGIF, they have a list of private contractors whom will do just that for you. So if you're interested, I've provided this link here for you all. which I think the PowerPoint will be posted for you all, so that you can get that if you need it. And lastly, I'll just mention that the Virginia Forest Landowners Education Program, which is run through Virginia Tech, is a really wonderful resource for landowners, and you can also find some resources there in terms of prescribed burning. And the head of that program is Jennifer Gagnon, and that's her email there if you'd like to email her as well for resources and support on that. And with that, I will whiz through my acknowledgements for my Virginia work and my Georgia work with the burning. And I will leave you with some fun photos that I've taken over the years doing field work. And if we have time for questions, I don't know if folks have other things to run off to do, but I'm happy to stay late if others are. So.
Well, thank you, Dr. Gleim. I really appreciate it. Really informative presentation. We do have a lot of questions, but unfortunately, we're probably going to have to just maybe, if you could just take 1 minute or some, I'm looking for themes in the questions. And there were a lot of interest in alpha-gal syndrome.
Mm-hmm.
And I was wondering, just as a closing kind of thing, if you could just say a few things about that as far as the causative agent or the vector, and maybe any research that you're aware of into that, because people have commented on here that they've seen a lot of that in Central Virginia.
Yeah, so alpha-gal, which sometimes is maybe also known as like the meat allergy, is associated with the bite of the Lone Star tick, which of course isn't good because that's the most common tick here in the eastern U.S. It's an allergy though, so it's not a disease or an infectious disease, right? So it's not caused by a pathogen, but it's simply if a person is allergic to a protein that the Lone Star tick will secrete or sort of, I guess, yes, secrete into you when they're biting you. So kind of similar to any other allergies, right? Some people may be bitten by Lone Star ticks and never have any problems with the allergy. Others, for whatever reason, may develop this allergy after they've been bitten by the ticks. Because it's not an infectious disease, I don't personally do research on alpha-gal, but there's certainly a lot of research going on into that particular allergy. And I was trying to think if there are any other specific questions about alpha-gal. I'm happy. And I guess the only other thing I'll just mention is a lot of times it's referred to as like a red meat allergy, and technically it causes you to be allergic to any mammalian meat. So, you know, that includes obviously red meat, but also pork and things like that as well. Deer, venison.
Great. Well, one last thing I noticed in the theme here, and then we'll end, is people are saying, well, you know, if dogs have a vaccine, why don't humans for Lyme disease?
That's a great question. And actually there was a human vaccine on the market back in the 90s. I don't know if anyone remembers that, but it was pulled from the market. And in all honesty, the official reason was that there were sort of negative side effects associated with the vaccine. But there's been some other sort of theories as to why it was pulled as well. In short, it's a great question. Why don't we have a vaccine? I don't know that I have a wonderful answer for you, but you're right. Obviously that it's certainly something that's being researched. I can say that for sure. But is something close to coming to market? No, not that I'm aware of.
All right. Well, wonderful. Again, thank you so much, Liz, for being here and just presenting a lot of information. Obviously, this is a very relevant topic because there's so many questions. And I apologize to everybody that we're not able to get to them today, but we do have your contact information. So you might get some master gardeners that are out there contacting you about a lot of things they want to know about.
Sure, yeah, please. If you have any questions, I'm more than happy to email with you. So feel free to shoot me an email and I'll be happy to respond.
All right, and thank you everybody for coming today and attending the presentation. I hope you've had a good Master Gardener college in general. And again, thank you, Liz, for all of the information. All right, everybody, we're going to end on that note. Thank you and goodbye.
Thanks.